B cells originate and mature in the bone marrow. The mature naive B cells have B cell receptors for recognizing specific antigens. These mature B cells leave the bone marrow, and they recirculate between the blood, the secondary lymphoid tissues, and the lymph. When these naive B cells encounter their specific antigens, they get activated and differentiate into antibody-producing plasma cells and memory B cells. There are two ways by which a B cell can get activated. These are T-cell independent activation and T-cell dependent activation. This classification is based on the type of antigen encountered by the B cells. Let's understand this further. In the previous video lecture, we studied that B cell signaling requires clustering or cross-linking of B cell receptors on antigen recognition. This clustering of B cell receptors is required to activate the accessory proteins Ig-alpha and Ig-beta. The antigen binding signal is then conveyed to the nucleus. This clustering of B cell receptors depends on the type of antigen encountered. For example, this antigen is having multiple repeating epitopes that can directly cross-link B cell receptors. Such antigens can activate B cells to produce antibodies without T cell help. Examples of such antigens are polysaccharides, glycolipids, and nucleic acids. Therefore, the antigens, which can trigger B cell activation without T cell help, are called T-independent or thymus-independent antigens. And this B cell activation, without T cell help, is known as T-independent B cell activation. But most of the antigens are proteins in their chemical nature. They form the largest group of antigens, but they do not contain multiple repeating units. This makes the cross-linking of B cell receptors difficult. So, when a B cell encounters protein antigens, T cell help is required to trigger B cell activation. Antigens that trigger B cell activation with the help of T helper cells are known as T-dependent or thymus-dependent antigens. And B cell activation which requires T cell help is known as T-dependent B cell activation. In this video lecture we'll study T-independent B cell activation. Let's say this is a T-independent antigen. This means it is multivalent. It is composed of repetitive and identical epitopes. B cell recognizes and binds to this antigen. Clustering of B cell receptors triggers the B cell activation. This is the first signal for T-independent activation. To get fully activated, these B cells require a second signal. This signal can be derived from other molecules present on the antigen. For example, B cells also have toll-like receptors that can recognize the various microbial surface molecules. This recognition and binding to the antigen by toll-like receptors generates second signal for T-independent B cell activation. After activation, these B cells differentiate into plasma cells that mainly secrete IgM antibodies. Memory cells are not produced, or if produced, they are very less. This is because memory cell production requires T cell help. So, this was T-independent B cell activation. In the next video lecture, we will study in detail the T-dependent B cell activation.